Hi everyone, welcome back. So previously we explored our innate immune system, our really diverse collection of tactics and defenses that we have to protect the body from pathogens immediately upon infection. So as soon as we realize that something is here that doesn't belong, we initiate an attack against it. All of those methods, though, are non-specific. They're search and destroy, but they're not looking for a specific target. They're looking for anything that doesn't belong. There's a bunch of issues with this method. It's not as effective sometimes as like a very targeted search, and there are more effective, more fast-acting defenses and a more coordinated effort that is sort of locked behind our adaptive response. So this is a process that really needs to be initiated. Once we learn what is attacking us, like the identity of the attacker, the pathogen that's infecting us, then we can take that information, feed it to the cells responsible for our adaptive response, and craft a very coordinated, targeted effort to eliminate the thing that is infecting us, the specific thing that is infecting us, okay? So I like to think about, as we talk about this, I like to think about our innate defenses kind of like beat cops that are out and about, they're patrolling, they're looking for potential crime. This is like, you're not looking for anything specific, but if there is a burglary in the area and you get the call, those beat cops are going to be the first to show up. They don't know who broke in. They don't know who is the criminal here, but they're here to address the problem, and they're going to look for individuals that are suspicious, right? The adaptive immune system is so much more targeted. It's almost more like the FBI, where you have specific criminals that you are looking for. So like, here's a wanted poster for like these the most wanted hackers. Here's a big reward over here. This is because they have the information that says these are the culprits to specific crimes. And so they're creating a very coordinated effort now to hunt down and, in this case, capture, but in our immune system, kill these bad actors. So how do we do that? Well, some phagocytes have this ability to talk to the adaptive immune system. In particular, like our dendritic cells are really responsible for this. So they have this, when they swallow something, something foreign, something that is causing trouble, some pathogen, they break it down in their lysosome. And then they take the pieces of that thing and they will bring it to the surface of their body. When they break these things down, they make them into antigens. Okay, an antigen is just some material that is recognizable by the immune system, that the immune system could target. Okay, so by taking, say, a bacterial cell and ripping it apart, they take the pieces of that bacterial cell, things mainly that are on the surface of that cell, and they attach it to these special proteins that are on the outside of the phagocyte. Okay, these are called the major histocompatibility complex proteins. Okay, otherwise known as the MHC. All right, you don't have to remember the whole name for this. Just make sure that if you see major histocompatibility complex, that's what we're talking about here, the MHC. These are special proteins that adorn the outside of a phagocyte. Their job is to hold on to an antigen. And this is essentially, this is now show and tell. Okay? So the antigens are placed onto the MHC proteins, and the phagocyte will take the antigen and bring it around to all of these different adaptive immune cells and show it to them. And present it to them like, hey, do you recognize this? Do you recognize this thing? Okay, and that's just being displayed right here in this image. So you have an antigen that is brought into this antigen-possessing cell. This is a phagocyte that has swallowed something and is broken it down. Then it attaches it to those MHC proteins. Okay, and then it will follow, like track down and bind to, or try to bind to a cell that's sensitive to that antigen. It has a receptor on it that can actually bind to that thing. Okay, so when we have an MHC molecule that has an antigen on it, the phagocyte will try to find a cell with a matching receptor. Okay, this is also what a major part of the lymphatic system is. It's kind of a highway that these white blood cells can travel and then find each other and have this sort of communication. Okay, so these phagocytes, when they do this, they look for a cell with the specific receptor that matches their antigen. The really interesting thing here is that these antigen receptors on the cells that have them are insanely variable, insanely diverse. So this is part of what takes so long for our adaptive immune system to activate, because these phagocytes are traveling through our lymphatic system, meeting up with every cell that has these antigens and trying to find one. It has these receptors for these antigens and trying to find one that matches the antigen that it has captured. As it does, a lot of them won't match. And that's because as these cells are produced, they have a really unique method of genetic recombination where they take the genes that are responsible for making these antigen receptors and they will shuffle them around really aggressively, like a lot. And as a result, they produce these constantly novel receptors to, in theory, to that match with potential pathogens that we have never encountered. So, in theory, there's a capacity for us to have a T cell with the antigen receptors that match literally any pathogen that we could encounter. So really cool. Doesn't always work out like that. It can take a long time for this to take place, and we don't necessarily have every single possible iteration of these antigen receptors, in our body at one time. But it is something that does happen here, and most of the time this is successful. So when you have an APC that comes and finds a cell that has a receptor that does not match the antigen, the information is not communicated. If there is a match, a bond will form, and that will initiate a full response. That begins the actual initiation of our adaptive immune response. These APCs, they communicate with what we call helper T cells. Okay, helper T cells. And these helper Ts are used to actually initiate that response. Sometimes we call those CD4 cells because of the name of the receptor that they have, those antigen receptors. They're called CD4 co-receptors. But so you might see them written as CD4 cells or hear them talked about as that as well. But I call them helper Ts. When you have a helper T cell that becomes active, it will start to engage other adaptive immune cells. It'll go to find other T cells of different kinds, we'll talk about those, and also B cells mainly. And those are the things that are immediately engaged. Okay, and we'll talk about each of those in a moment. The information, the identity of that pathogen that is infecting you is relayed. And so all the cells that are awakened, that are activated by this helper T, are activated with the target in mind, that they are going to be formulating now an effective attack against this specific antigen. the specific antigen that is in theory attached to the pathogen. So one of the first things that are activated are those B cells, okay? And these are the antibody-producing cells. Now, there's two kinds that we talk about. There's the plasma cells. Those are the ones that actually are actively making antibodies. And then we also have the memory Bs, okay, memory B cells. These are responsible for holding on to the recipe for a specific antibody from a past immune response. So, for example, if you've had measles before, for example, or more likely, you've had the vaccine for measles, you are walking around here with memory cells, these memory B cells that remember the recipe for an antibody that binds to a specific antigen on the measles virus. These antibodies are really cool. They have a variety of like impacts. Their roles are kind of diverse, but one of the big things they do in their ability to fight pathogens is that when they find something that they match with, they will do something called agglutination. And this is essentially when an antibody finds its matching antigen, it binds to it, and then it will bind to other things that also have that antigen, okay? Which is usually other cells or other particles, things like that. And they will start to clump together, okay? They will literally like bind up, clump up, and act as both immobilizing agents, where they sort of stop the cell from doing its thing, from moving around, from causing some trouble, from interacting with other body parts, things like that, other tissues, and it also acts as a signal that can kind of flag phagocytes to come and help them clean up these bacteria. So it's a very specific method of attack. Antibodies are also really tough. They're kind of made to last. So when we make these antibodies, they can survive in the blood for a long time, months to years sometimes. And again, you have these specialized B cells that will continue to produce these antibodies, at least in small quantities, for a long time, even after that infection has passed. Antibodies are also really resistant to digestion, which is kind of crazy. So this is pretty cool because it means that you can also absorb antibodies through your digestive system. They don't get broken down by the enzymes and the stomach acid and other things like that as it enters your digestive system. This is a big deal for infants because when we're born, we have really poor immunity. Our immune system is what we call naive. It's uninformed. We haven't encountered all of the the different microbes at that time that we had gotten sick with over the course of our lives, and have developed this robust collection of memory cells that help us fight off that stuff that doesn't exist. And so antibodies are really important in infancy to help keep a child from being chronically sick and keep them healthy. And one of the ways that is conveyed is through an oral route. That by drinking breast milk, that the antibodies will be secreted into the breast milk and then be digested and absorbed through the diet of the baby, and so they can acquire it that way, which is really neat. So, of course, later on, these antibodies that are dietary can be replaced by the ones the baby makes as they begin to mount their own defenses. So these antibodies, when they bind to something, they can actually help target the immune system, start cleaning up something that might be infecting them and things like that, and that can create the conditions to activate the adaptive immune system, and then create those memory cells and so on. This is also something that can be used as a treatment for some diseases as well. So we can actually give somebody antibodies to specific diseases, and that can be itself a treatment for those diseases. So, there's lots of things like this. The treatments for botulism, diphtheria, hepatitis A, B, measles, rabies, tetanus, even like, even smallpox, although thankfully this one is not nearly as common anymore, since this is an extinct disease that we don't really have the need to do much treatment for this. But these are all things that we could do. We could give somebody antibodies, and even if they don't have the immune memory to fight this thing off, we can give them antibodies to help them do that right away. So then we have T cells. Okay, T cells are the other major kind of these are all white blood cells, by the way, that are part of our adaptive immune system. Okay, so these actually originate from lymphocytes. T cells and B cells both. They both arose from lymphocytes, and then differentiated based on where they matured. These are special cells that, they're all created in the bone marrow from bone marrow stem cells. But T cells, unlike B cells, went to the thymus, which is a small gland in your chest, to mature. These T cells, they end up remaining dormant until they're presented with an antigen, usually, by a macrophage, by one of these, or a dendritic cell, or by a B cell. They can also act as an antigen presenting cell. Now, these B and T cells, they differ a lot, not just by function, but also by like the targets that they focus on. Okay, so B cells, really good at targeting intercellular threats. Not within a cell, but between cells, in like the spaces within your tissues. So pathogens that hang out among cells can be cleaned up really effectively by B cell activity. Particularly like bacteria and fungi and parasites. T cells, however, are really good at dealing with like intracellular threats, as in like things that live inside of cells, viruses and some bacteria especially. Okay, together in concert, they're very helpful, but this is very important, a difference in their main function. So types of T cells. Okay, we have three. We've already talked about one, the helper T cells. Their main jobs are to receive the information for a pathogen that's infecting you. So again, an APC will come and bind to the surface of a helper T, and they will activate it, and it will do two things. It will secrete cytokines, which are these important proteins, collection of different small proteins that will stimulate a lot of other immune responses, including inflammation, but also activate other immune cells and help kind of direct them. And then they will also go out and actually communicate and activate these other T cells and B cells, and also these macrophages that will start getting involved more directly. So they'll be able to start looking for something specific. You also have cytotoxic T cells. These are called killer T cells also. So these are like very direct search and destroy, attack and destroy for these abnormally behaving cells. This includes foreign cells, but it can also be our own cells that are misbehaving. Oftentimes, our killer T cells are very involved in helping us clean up cancer. Memory T cells are the T cell variant of the memory B cell. They remember past infections and our responses to them. And so, likewise, if we ever re-encounter the pathogen that attacked us, we can reactivate these memory T's and memory B's and, have effectively a much faster response than we did the first time. And to that end, we have two major differences in the responses that we have to infection, okay? And these differ by like when we encountered them. Like, is this our first encounter or our second or third or fourth or fifth, blah, blah, blah. All right? Primary immune response. This is our first exposure to a pathogen. First time we were exposed is a slow response. It takes a long time. There's a lag period for our adaptive immune system to be activated. Usually this is like 3 to 6 days for the first antibodies to start to be produced, and it really peaks after 10 to 12 days, although there's a lot of variation. There's a lot of variation here, okay? Now remember, our innate immune system is active right away. As soon as we kind of encounter this pathogen, it starts to act. But to activate this adaptive immune response, it can take much longer in this case. The secondary immune response is any secondary encounter with a pathogen that we've been exposed to before. So you contracted the measles. You got over it. You fought it. You know, it was really bad, I'm sure. It'd be really bad for anyone to get the measles. But then you encounter it again later, maybe like 3 years later, let's say, and you have this robust immune response now to measles. And so, it is much faster. The lag time is hours, not days. And the peak antibody production is much sooner as well. It peaks in just a few days. And another kicker is that you have a much more significant amount of antibodies that are being produced. So the big takeaway here is that your secondary immune response is very fast and very intense as well. There's a massive surge in antibody production that dwarfs the amount that is produced in the primary immune response. Often it is the case that in your secondary immune response, you didn't even realize that you were sick because the infection was dealt with before it ever became a problem. So this is kind of what that looks like here, all right? So our first exposure is kind of short, right? It is like the antibodies that are produced and not as much, and it takes a while for that peak to happen. But at that second exposure, you get exposed again to that same thing. Say, influenza, some influenza A or whatever, some specific strain. Second exposure, third exposure, fourth, fifth, you have this huge spike that takes place, okay? And then a much slower tapering off. And so those antibodies, remember, you make those antibodies and they circulate in your bloodstream for a long time, months to years. And those memory T and B cells also last a long time, many years. And they can slowly wane. Our immune system doesn't last, those immune memories don't last forever. But it can last a long time, many, many years, all the same, okay? And so, that secondary response, very swift, very intense, all right? Importantly, if, in my previous example, if you had gotten the measles vaccine instead of contracting the measles, right, and suffering the measles as you did, that is also the primary immune response that takes place when your body responds to that vaccine. It just has the added benefit of not actually making you horribly, horribly sick or also infectious. So not only are you not sick and not infectious, not capable of spreading this to other people, but you get the same immune response at the end. Same significant immune memory is formed so that the next time that you get exposed to measles, this is your response here. It's fast and intense. Immunity is something that we build over time, over the course of our lives. Every single thing that we are exposed to that tries to infect us, we mount a response. And if we are successful, we survive the infection, we remember it. We remember that memory of that infection and how to fight it. And so this is really important because, over time we are building this. Through the exposure that we have to microbes in the environment, we build this process, this intense response, and this library of a memory. Our bodies, after each infection, we're going to be storing a few of these memory cells somewhere for us to go back to at some later point if we ever really need it. that doesn't mean that those cells live forever or that the immunity lasts forever. Immunity can wane, and some immunity lasts longer than others. Sometimes the intensity of an infection or like the significance of an immune response can influence the length of that immune memory. This can vary from like infection to infection as well, but it also can vary based on the the conditions of our lives. Through our aging and also our general health, we can lose immune function. We can have some parts of our immune system wane faster than others, or have issues. In this case, we can call somebody that experiences this, they're immunosuppressed or immunocompromised.